I'm struggling a little bit to judge my audience. I know that there's a lot of people in here that are international ag students. Others might be plant breeding students. So as we talk today, we may get a bit technical in some of the plant breeding stuff. For some of you, that will be a bore. For others, that will be the exciting part. But I will do my best to balance both of those things, to talk both about some of the technical work that we're doing, but also to highlight, I think, where that technical work has impact and where we feel like that technical work fits into a larger picture of development solutions that IRRI tries to support through their networks. The title of my talk is Transforming Rice Breeding. It sounds a bit presumptuous, but that's actually the name of the grant. We're primarily funded through the Bill and Melinda Gates Foundation on a grant right now called Transforming Rice Breeding, which the idea is to take traditional breeding programs that historically have struggled to integrate modern technology and modern strategies effectively and to modernize them. And I think part of the attraction that IRRI had, so part of the reason why I feel like I was competitive for this position at IRRI was because I have 3 years of experience working in industry. And in industry, efficiency is always the name of the game, and the kind of training that we receive in industry, technical training in terms of plant breeding, is quite intense. And so what the objective, I think, is to take some of that training and the intensity and the efficiency of that training and then bring that to the CG, and then from the CG bring that to national partners and see where there are opportunities for innovation, where there's opportunities for improvement. So just a brief outline, we'll go through the requisite situation analysis. Rice is important and feeds a lot of people. I want to talk a little bit about sustainable development just because I think the term itself is kind of interesting to me, and we have now these sustainable development goals that have succeeded the Millennium Development Goals. So this concept of sustainable development is interesting. We'll spend most of our time here where we'll talk about some of the work that we're doing in TRB, which we can consider a case study in sustainable development. And then we will finish up with some of the efforts that we've had to take what we're doing at IRRI and extend it beyond the borders of the CG into some of these national partners. So I won't spend a whole lot of time on this because I do think that at this audience especially, it's important that you guys understand this. In other venues I speak, they don't always. But rice is incredibly important. This is one of my favorite slides. I developed this while I was here, actually. But this map comes from the National Geographic 7 billion project. So when the 7th billionth person was born, National Geographic did this whole expose. And they produced this map, which I really love. And it's sort of, it's the brightness is the population density and the color is the income. And some of the things that I wanted to highlight on this map is this circle right here. More people live inside of that circle than outside of it. And then if you notice, I've overlaid on top of this map the percent of the calories from rice that people consume. And so when you notice right away is inside of this circle where more than 50% of humanity lives, it's awfully warm colored, which indicates that it's, there's poverty there. But then you also see that most of these countries have a very high consumption of rice. Rice is an important part of their diet. Rice is an important part of their economy. Rice is an important part of their revenue streams. Many people are, their primary source of revenue is agriculture, and rice agriculture in particular. So if you want to work in a crop that sort of directly impacts the food that people eat, rice is a beautiful crop to work in. When I was at Pioneer in particular, I would tease the maize breeders about that because nobody eats field corn except through beef. And so this was a fun slide for me to throw up at Pioneer often too. Situation analysis. So this is a lot of material that just comes directly lifted from IRRI, but I think everyone understands that we have to feed 9 billion people here coming up. Most of those people are going to live in Asia, and in addition to just sort of providing the requisite caloric intake, it's important that food be nutritious as well. And so we have obviously concern about children and stunting and other nutrition-related aspects. Some things that we don't often think about, though, when we talk about feeding 9 billion people, the landscape is changing dramatically. There's shifting roles in the public and private sectors, and particularly in rice, the private sector is taking a much stronger interest than they have historically. And that brings money, but that money is also targeted to specific things. It brings the development of public-private partnerships, which can be quite lucrative from a development point of view. The technological landscape has changed quite a bit, and that is also giving us opportunities and also giving us liabilities that we have to work through. So with that sort of precursor, you should be familiar with these. These are the Sustainable Development Goals that have been adopted by the UN. This entire framework, more or less, is a way to gather the funding of the world and focus it more intensely on specific problems so that people who are all engaged in finding solutions are able to harmonize their efforts a little bit more. And IRRI is no exception to that. So we are in the middle of a reorganization, for example, at the Institute, and part of the organizational units that we're creating, and many of us are leading, we do what we can to draw our activities back to these Sustainable Development Goals, because what this does is it makes it easy for those who are providing funding, granting agencies, who are accountable for making sure that the money that they're providing impacts these. If we can show them how what work we're doing connects to these goals, it makes it very easy for them to provide a funding mechanism. But that being said, what I wanted to focus on is this idea of sustainable development. So what does that even really mean? And when you think of the word sustainable, it's a buzzword that gets thrown around quite a bit, but more or less what it means is that something is able to exist in some level of perpetuity. So I've come up with my own little framework for trying to understand sustainable development, and that is this here. You've all heard this adage, right? So if you give a person a fish, they eat for a day. So for me, if you take it out of the local context and talk about it in the global context, if you give a person a fish, they have food security. They have food. If you teach a person how to fish, and I put responsibly in there because I think that's important, if you teach a person how to fish, then what they have is a durable framework for food security. It's not just food security. If you teach a person how to fish, that's when you have sustainable development, right? And so that for me is, I think, what we're trying to achieve in this grant at IRRI, in the breeding programs. It's not about providing rice varieties to the world. It is about that. We develop them. But it's also about modernizing and making efficient the rice breeding programs. It is about that. What I'm interested in is making sure that those modernization efforts can live in perpetuity, that we're training programs and not people. And so as people move through those programs, as people move on to other things or retire and there's attrition in the staff, we don't lose the innovations and the development that we've had. Those programs are trained and those programs are continually innovative and efficient. So this for me is sustainable development. If you teach a person to teach a person how to fish, then you have sustainable development. So let's dive in a little bit now to the work that we're doing specifically. I want to show you some of the stuff that we've been doing at IRRI recently. This slide comes from Gary Atlin, actually, who's at the Gates Foundation. But we have a problem in rice, and this is a problem that doesn't exist necessarily in other markets, but in rice, the farmers in Asia in particular that are growing rice, the varieties that they're growing are old. The average area weighted age of varieties in farmers' fields in Asia is 28 years. And that means that the material that they're growing now in their field was bred and selected really just right after the Green Revolution. And it begs the question, what is the impact of plant breeding today? Those of us who are engaged in this activity and constantly releasing new varieties, what is the barrier to adoption? Why are new varieties not being selected by these farmers? Also, this comes from a paper in 2012 in Nature Communications, but you can see the orange color here. They say yields have stagnated. Yields are not going up over time. The dark green colors here in Asia you see is where yields are moderately increasing. And so we see that not only are the varieties that the farmers growing old, but we're also not seeing an increase in yield per acre or yield per hectare. And this is a concern because as breeders, that's what our job is to create new varieties. Now, there's a million different reasons why a farmer's choice to grow one variety over another, million different variables that might influence that. But what we want to do is test some hypotheses, and one of the hypotheses that we wanted to test is, are breeding programs making genetic gain? Are we actually producing better varieties? If we are producing better varieties and they're still not getting to farmers' fields, that's a seed systems problem and needs to be addressed at the seed systems level. If we're not producing better varieties, then that's a breeding problem. I suspect the answer to the question is both, and we will eventually elucidate some of those over time. But what we want to focus on, because this is more or less the core of what I do, is the breeding component. So there has been efforts to estimate genetic gain in rice, and here's a couple of different papers from Xiaobing Peng, who used to work at IRRI, now works in China, attempting to measure genetic gain, and particularly in IRRI breeding programs here, and these are in Chinese breeding programs over time, they consistently come out with this number here. So this number 0.075, what that means is 75 kilograms per hectare per year. That's how much they feel like yield is increasing over time. In this Chinese study, he came to the same slope of that line. I don't particularly, from a scientific point of view, I don't particularly like these analyses. I think they're underpowered and they misrepresent the situation a little bit. Most of these are what we call IRRI trials, where you take really old varieties and you grow them in really modern environments, and you compare them to really modern varieties that have been selected to perform in really modern environments. And so I do think it distorts the genetic trend. Most of these are grossly underpowered. You get one location of data, maybe 2 seasons, coming from the private sector, where I wouldn't even think about releasing a variety unless I've grown it at thousands of locations and have extreme confidence in its ability to perform. I would never base a decision on that kind of information. Of course, realizing that you have to work with what you have in terms of funding, so you try to get to the best answers you can. This only represents released variety, so everything that's created in the breeding program and rejected is not included in these analyses. And so it's a little bit biased too from that perspective. And they regress this on the year of release. So these years here are the year the variety was released, not the year the cross was made. And there's a lot of things that there's a lot of bureaucracy in the system that either delays or prevents the release of a variety, where the year here indicated may not necessarily be the best year to associate with that variety. So what we decided to do, since I have a breeding program at IRRI, is we decided to go into the historical data of our breeding program. And so I went to the last 5 years of data, where we have 102 trials and 17,000 lines total. Those lines have been grown in over 23 different environments, and what we wanted to do was model more or less the performance of those lines over time. I won't go, given the nature of the audience, I won't go too much into the technical details of how we constructed that model, but if you're curious, we can talk about it. But one of the important things that we did here is we used pedigree relationships between these lines in the model, which means it allows us to say if a particular line is in the trial, we get a value for that line. If the relatives of that line, cousins, first cousins, parents, sons, daughters, if the relatives of that line are also in the trial, that gives us information about that line, because we know information about its genome and how its relatives are performing. So having that as the relationship matrix has helped a lot, and we created these predictors. So if you look at the breeding values that we've created based on this analysis and regress them on the year that the cross was made, not the year the variety was released, what we see is from 1960 to 2014, this genetic trend is not the nice straight upward line that some publications would have you believe, but is actually not even really linear. There are periods of time when we are making genetic gain. Yields are going up. Genetic value is going up. There are periods of time when genetic value is going down. And if you take, this is not a linear trend, but if you bastardize the statistics and try to put a straight line through there, you find out that the trend is not very high. Did I put it in here? I may not have. I think the trend is about 13 kilos per hectare per year is the overall genetic trend since 1960. And that's not a very flattering estimate, because when you look at modern day corn breeding programs or soybean breeding programs or some of these other programs that come from the private sector, they're consistently making a much higher rate of gain. And we believe that there are inefficiencies in the breeding systems that are driving this. And so anyway, so this is the genetic trend over time. The question then becomes, are we making gain at all? Like, is the breeding working? And so if you consider a breeding cycle, and for those of you who aren't plant breeders, a breeding cycle is the time from the day I make a cross and create a new line until the day I use that new line as a parent in a new cross. That's a one full breeding cycle. And in theory, over the course of that cycle, I should be making gain. The parents that I choose should be better than the parents I had before. So we went into the pedigrees and we figured out the depth of the pedigrees, and for every single line we mapped out how many full breeding cycles had occurred before the creation of that line, and then we regressed the breeding values on that. And it turns out that we do get a reasonably linear trend of about 92 kilograms per hectare per cycle coming out of the breeding program. So we are making gain. We are measuring yield effectively. Every time we complete a breeding cycle, we are creating better lines than what we had before. The problem is, our breeding cycles are incredibly long and variable in length. And so when we take the equivalent complete generation here, which is more or less the breeding cycle, and regress it on time, what we see is that in some periods we were able to create parents and then use them really quickly and create new lines again. And then there are other periods of time when we spend 10, 20, in this case 30 years never advancing the breeding cycle, but always working with the germplasm within that one cycle. And that, I think, has been the primary driver of the slope of that genetic gain, why we think it's not been very high. And so that's good, because a well-defined problem is half solved. So now we can begin to innovate around that and say, what can we do to make the breeding program more efficient? This is IRRI's breeding program, the one that I run at IRRI. I would be very hard-pressed to find that this pattern would be any different in any other rice breeding program in the public sector. People may prove me wrong on that, but I don't think it is. So part of the challenge here, I think, is it's been a lack of focus on what I consider to be the quantitative components of a breeding program. So we talk about the modern synthesis. Those of you who are science historians will recognize that the modern synthesis occurred in the early 20th century. This is the reconciliation of Darwinian evolution with Mendelian genetics. And we started to finally have a mechanism for descent with modification that Darwin proposed. And I often use this as a type to say, hey, on this side, on the Mendelian side, it's very common to take genetics, to take genotypes, to grow them in environments, to map QTLs, to figure out that those QTLs have impacts on the phenotype. And then once you have all of these genes in the genome that you believe have an impact on the phenotype, you stack them all together in the same genome, and you create a line that has a decidedly different phenotype than the line you started with. And that's a very Mendelian approach to plant improvement, variety improvement. I believe that this side of the equation, where you grow a bunch of lines in environments, they have phenotypes, those selection acts on those phenotypes to influence the frequency of alleles in your gene pool, that side of the equation has been neglected in a lot of ways. And what we want to do is beef up our strength here without losing our strength here to create a more integrated approach to plant breeding that allows us to leverage both of these mechanisms. But yeah, so these are two distinct trait deployment strategies you want to consider. So what does the landscape look like? The landscape has changed quite a bit. If we want to really beef up our strength on the blue side of that equation, we need to use this here. So this is probably the most technical we'll get in the presentation, but this is the breeder's equation. If you don't know what it is, I would suggest you learn it. Even if you're not a plant breeder, if you are a plant breeder, I require that you learn it, but if you're not a plant breeder, it's worth knowing anyway. But what this equation is it's basically an equation that predicts the rate at which your phenotypes are going to change over time. And there are variables in this equation that we can manipulate in order to increase or decrease that rate. But over time, the scientific landscape within which we've been breeding has changed a lot. And so I put a couple of token examples here, but in the 1960s is when the Green Revolution started and all of the rice became semi-dwarf. In the 1970s, there was a lot of work on statistics and experimental design that actually I think we forgot a little bit in the 80s and 90s, and I keep going back to. I've got these wonderful old documents at IRRI that I consistently go back to when I talk to the team about what plot size should we use, what design should we use in our experiments. A lot of that work's been done in the 70s. In the 80s, we started to see molecular biology take a lead role. Also, quantitative genetics came into its own in the 80s. In the 1990s, there was a lot of QTL mapping and gene discovery. The 2000s brought genomics. And then lately in the 10s, we've seen a lot of omics technologies, high throughput phenotyping technologies that have come out. So the scientific landscape has changed. And what we need to do, I think, in order to strengthen our capacity here is not get distracted by the technology, by the changes, but rather to think pragmatically about what is available to us now that we can do, that we couldn't do before, when we didn't have access to these kinds of technologies. But the technologies themselves, the pursuit of high throughput phenotyping, the pursuit of genomics does not create new varieties. The integration of these tools to help us manipulate these variables in the breeder's equation, that's what's going to create new varieties. And so that's what we try to do. So what can we do here that will help us influence here? So I'll give you a little introduction into this equation. The first thing I think we can do here, this sigma A is our genetic variation. The first thing we can do is start to manage our genetic variation a little bit more closely and a little bit more carefully, and we'll talk about how we're doing that. And one of the primary ways in which we'll try to do that is elite by elite crosses. We can do faster breeding cycles, so that's why here in the denominator. The longer those breeding cycles are, the slower the rate of genetic gain. So if we can make those breeding cycles go faster, we'll make more gain over time. We can test more varieties, right? So as we increase our selection intensity, which is I in this equation, we can test more lines. When you test more lines, you sample from that genetic distribution more fully, and you can find better material that you can move forward. And the way to test more lines is by reducing costs, because my budget's not going up, And so if I need to test more material for the same amount of money, I need to reduce costs. And then the last is making more accurate selections. So that's R in the breeder's equation, is selection accuracy, which means we need to make sure that we have decent heritability. We need to be able to measure plants and know that the measurement we're making is a measure of the genetic quality of that plant and not some other non-genetic influence on why that plant might look good or perform well. And so we're going to go through each of these one by one. And end up, what time do we end so that I hit the right? 1:10. Okay, so we've got plenty of time. So I'll show you a little bit about what we're doing at IRRI. So this is more figuring out how to fish. Okay, so we want to give a man a fish, but we need to figure out how to fish ourselves. And so we're trying to figure out how to fish ourselves, and then we'll go into a little bit about what we're doing to try to teach others how to fish, and then we'll speculate a little bit about how we can teach others to teach others how to fish. So elite by elite crosses and population improvement. So IRRI is probably most famous for the tremendous genetic resource that we have, known as the IRRI Gene Bank. The IRRI Gene Bank is 127,000 unique landrace varieties of rice that comprehensively survey as much as we can, all of the genetic diversity that we have in the species. And that resource, which is represented in yellow here, is studied extensively throughout the world. And people are always talking about unlocking that resource, leveraging the genetic diversity in that resource, using that resource to advance breeding. I like to make the argument that there's actually a second equally valuable genetic resource at IRRI that is not the gene bank. And that is the rice that actually feeds the world. That is the rice that represents the elite breeding germplasm that we're actively working with, that our partners are actively working with, and that is improved for its agronomic and quality. So what you see here is the white. So this is just genotype data that's been put on a PCA, but this white material is the breeding program, and this yellow material is the gene bank. And not surprisingly, they're decidedly different. And they have different qualities. So the primary driver of genetic variation here is mutation. There's a lot of allelic diversity in this space. In the breeding program, there's not, or there's much less. And so the primary driver of our variation is recombination and crossing. This gene pool is static, more or less. We attempt to make it as static as possible so we don't lose anything. This gene pool is dynamic. It's constantly changing. We capture value from this by looking for rare alleles that have really large effect that we can then pull out and use in the breeding program. We capture value in this by enriching the frequencies of elite haplotypes over time through recombination. So I like to try and give this more of an identity than it currently has so that we can begin to put the kind of intellectual input into the elite breeding germplasm that people typically put into the diverse germplasm. So this is our galaxy. I call this a galaxy because I wanted to be an astronomer when I grew up, and I ended up as a biologist. So I have to put some astronomical references in there somewhere. So this is our elite breeding germplasm. This is that same analysis that we just did. There's 17,216 lines. And you can see the distribution of our breeding values for yield here. The average yield in our galaxy is 4.4 tons, and it has a genetic standard deviation of about 0.4 tons. So there's the variance here. With this elite resource that we're trying to put a fence around, we're trying to give an identity to, we have a mandate from our funding sources to make genetic gain in the rate of 1.3% gain for yield per year in the breeding program. We're currently making about 0.3%. So we need to step up and increase the slope of that line like we showed you. And the way we do that, of course, is through what we call recurrent selection. And the idea is that we take our current galaxy, we select the best of the best, we allow those best to intermate with each other, they have children, we evaluate their children, we select the best again, and then we let their children intermate with each other, which I think is sometimes where breeders step off. I think breeders are afraid that they're going to run out of genetic variation if they do that. So they're constantly taking those children and crossing them to wild, it's not wild rice in the sense that Susan would call me out on that, but wonkadonk rice that comes from the gene bank that is not agronomically improved. Or, even worse, they'll cross back to previously released varieties that have had a history of doing well. And the problem with that is if you imagine genetic gain in your program like a ship that's sailing out to sea, and you're constantly crossing back to these old varieties, it's like dragging an anchor on the bottom of the ocean, and it's slowing your ship down. And so what we want to do is focus on recombining the genetics that are in this space that we're selecting. As you do that, what happens is these guys become the parents of this generation, these guys become the parents of this generation. Over time, your diversity narrows, but over time, your means also increase. And that's an important principle on the blue side of that equation that I think rice breeders that I interact with often miss, and we try to emphasize. Why do we believe this works? We believe this works because it's just evolution. It is the commandeering of evolutionary principles that occur naturally on the planet for the purposes of plant breeding. So these are Darwin's finches. And if you're familiar with the HMS Beagle and the voyages of Darwin, he went to these Galapagos Islands and he saw tremendous variation in the finch populations. And so what they speculate happened was, on the mainland, there was a hurricane that blew a bunch of finches out to the Galapagos Islands. And then those finches began to intermate with each other. And there were selection pressures that were applied to those populations. One of the stories you get is that the finches with short, fat beaks could break the seeds more easily and they could then eat better and then reproduce better. And so from the relatively small diversity of finches that were blown to the island, and then selection was imposed on their populations, you get a tremendous array of different phenotypes. And it wasn't because new finches with short, fat beaks were blown in from the mainland that were more successful than the finches that were blown in the first time. These guys are the descendants of these guys, even though they didn't have any short, fat beaks to start with. And that's another message that I try to drive home. If in our galaxy of elite lines, we may have a low average, we may not be high yielding, or if it's another trait, we may not have high grain zinc concentration. But as long as we have a variance, we can act on that variance, and we can improve that mean over time. Whereas I think people sometimes assume if the mean is low, I immediately need to go and introduce novel alleles that we don't already have to help improve that mean. I argue that you can improve the mean without doing that. So this is just evolutionary biology at the end of the day. Here's a little bit of a mechanism as to why we think that works. So we'll get a little bit more technical here, but again, it's important, I think, to understand. This is an example in human height. Okay, so we've been studying human height for a long, long time. And not surprisingly, human height is distributed continuously in the human population. They have done tremendous studies in the human community to map genes associated with human height. They've had populations of greater than 10,000 individuals and have mapped over 50 different regions of the genome that significantly impact human height. And yet, when you take all of those QTL together, you're still only explaining about 5% of the variation in the human height phenotype. So you're not explaining a lot of the variability, even though you found as many significant genes as you could. This paper here from 2010, Yang et al., used a genomic selection model, and what that basically means is they used all of the genome, not just the significant parts of the genome, to describe human height, and they were able to explain 45% of the variation instead of 5%. And when you look at this, so this is a paper that came out recently out of Jonathan Pritchard's lab, who's a famous human geneticist, and in this figure he says, okay, here are all of the markers in the genome that are surveyed. Each of them has a significance level in terms of how much of an impact it has on human height. If you look at the markers that are highly significant, you find that most of them have an impact of about 1.4 millimeters on human height, which isn't surprising. But then if you look at all of the SNPs together, including the ones that are non-significant, any individual marker will have an impact, an effect size that's about 10 times smaller than that. But in this paper, he proposes what he calls the omnigenic model of inheritance, which poses this hypothesis that says, what if all genes had an impact on all phenotypes, even if that impact is infinitesimally small? What if, rather than looking at the genome as this reductionist, every gene comes together and has its own individual impact on human height, what if phenotypes are an emergent property of a complex biological system? And what we can do is manipulate that system to increase the probabilities, but we can't control directly what the phenotype ends up being. In this omnigenic model, the idea is that everything's important. And in aggregate, all of these regions of the genome that are not significant actually have a larger impact on the phenotype than all of the significant ones put together. And so the only way to capture this, you won't capture this through genetics by identifying every single one of those and then trying to stack them together. The only way to capture this is by evolution, by acting on the phenotype. And there are ways in which we can advance that through genomic selection and other things. But the only way to really truly measure what the concentration of favorable alleles is within any given organism is to accurately measure its phenotype. So some people argue, and this is probably the primary argument I get, they say, Josh, if you put a fence around the elite breeding germplasm, and you only cross within that fence, and you introduce things in a very managed way, it's not that there isn't a gate there. There is a gate, but it has a really big padlock on it, and I'm the only one that has the key. And they have to convince me to cross with something if they want me to put it into the breeding program. If you do that, you will run out of genetic variation. And what will happen is you will stop making gain over time, you'll hit a yield plateau, and you won't be able to continue. And that's a valid argument. So what we did is we took our breeding program and we simulated it using BSL, actually. And in our breeding program, in our galaxy, we have an effective population size of about 33. So we simulated effective population sizes of 10, 20, 50, and 100, given the breeding strategy that we currently have set up. And we simulated how many breeding cycles can we do before we start to hit a plateau. We quickly realized that we didn't have the computational power to do that before this seminar, so we limited it to 20 breeding cycles and asked the question, how far do we get out when we do 20 breeding cycles? And it turns out, actually, right up until about cycle 10, we have a pretty linear relationship with genetic standard deviation in cycle time, which means, remember, the standard deviation in our program is 0.4 tons of rice per hectare. For every breeding cycle, we advance about 0.4 tons. That's the prediction. And so once we hit breeding cycle 10, then you start to see it starts to inflect a little bit, and then around 20, we start to see maybe there is an inflection point there. And as we simulate more and as we go out further, we'll see how much of an inflection point we have. But the thing I want to emphasize is that it takes us 4 years to accomplish one breeding cycle. So if I have 20 breeding cycles worth of time here, we're talking about 80 years of plant breeding ahead of us. And I refuse to not make progress for 80 years on the off chance that I might run out of genetic variation. I would rather make progress during that time and then deal with the problems that arise as they arise and monitor our germplasm as it moves forward. So that's the blue side of the equation right there. And that's elite by elite crosses. And that's one of the big changes I think that's coming to IRRI's breeding program, and also that we're trying to distribute to the rest of the breeding community. I'm going to combine these two together because we use a similar strategy, but what we want to do is test more recombinants at a cheaper price, and we want to make our breeding cycles faster. So the breeding cycles have historically been about 10 years, and we want to shorten them to 4 years. That's where we're headed. So this is how, like a traditional rice breeding program, this is how it's set up. And I won't go into the details here because if you're not plant breeding students, this may not be interesting. But what this is called is pedigree selection. And there's a few problems with pedigree selection that I take issue with. The first is that we do single plant selections of segregating material. So if you put an F2 population in the field, every single one of those guys is a different genotype. But they're not true breeding yet. So all of their children are also going to be different genotypes, and their grandchildren are going to be different genotypes. And what we want to do is get them to the point where they're true breeding, where one generation and the next are the same, and then evaluate them at that point. So if you're looking at single plants at one location in the field and all of them have a different genotype, and you say, oh, look at this one. I'm not kidding you. I've had breeders put the panicle in my hand and say, you can feel it, can't you? No, I can't feel it. You look at, I mean, it's a beauty contest often for these plants. And so you see the ones that look the prettiest, but you don't know if that was planted in a pile of dung, and that's why the plant looks really nice. Maybe it's the most heterozygous plant in the field and it most closely resembles a hybrid, and that's why it looks really good. It's not necessarily because the genetic value is high. So that's one issue here. The breeding cycle is 11 years. Like before you get down to a fixed line and can begin to consider using one of these guys as a parent in a new cross, it's 11 years later. This takes hectares and hectares and hectares of research land. We're planting F2s and F3s and F4s out on all these lands, and that's expensive. Often it's only at one location, so there's no estimate of genotype bioenvironment interactions, and there's a really heavy investment in very early generation breeding material. And what we want to do is flip that around on its head and make a heavy investment in the lines that we believe have a higher probability of succeeding. So one of the things that we did was we developed this rapid generation advanced greenhouse. So rather than planting F2s and F3s and F4s and looking at them, all we're trying to do is go from F2 to F3 to F4 to F5 as fast as humanly possible. Now in the corn community, they've solved this using doubled haploid technology. Doubled haploid technology does not work as well in rice as it does in corn, not yet. But rice has an advantage. It's naturally self-pollinating. Rice sells for free. You have to pay money if you want to self corn. And so what we've done is we've created this greenhouse where we just allow the plants to self. We grow them in tiny little pots. It accelerates their flowering time. We can get 4 generations of rice in 1 year in the greenhouse. So we go from F2 to F6 in 1 year, whereas before it used to take 7 years to go to F6, right? So that's a significant acceleration of the breeding cycle. So here we go. We have our parents. We have a cross. Then this is all of year 3. is in this picture, and then by year 4, we're looking at amplifying seed. By year 5, we have them in a yield trial, and we're beginning to evaluate the quality of these lines and throw them away or keep them as well as they perform. It's much cheaper, the breeding cycle is faster, we impose selection on fixed lines. In this yield trial, we can do multiple locations, so we get to look at G by E, and we're investing more heavily in fixed lines that we believe are going to be successful. In addition to making the cycle faster, we also want to make it cheaper. So we have a strong collaboration with the Bangladesh Rice Research Institute. This is something that they started that IRRI is picking up on. We do it in a big, expensive, fancy glass house, and all of our national partners say, well, geez, IRRI, that's great, but I don't have a big, fancy, expensive glass house, and my boss isn't going to let me buy one. And so IRRI decided that they said, I bet I can do it outside. And I said, I bet you can do it outside. Let's see how it goes. And so they start doing it outside, and what happens? Not only can they do it, it's orders of magnitude cheaper than doing it in the greenhouse. There's tremendous advantages. So this is just back of the napkin math, where it cost me about 94 cents to take a line from an F2 to an F6 in my greenhouse. By doing it outside, I think I can get that cost down to 36 cents. Based on the experiments we're conducting this season that I've seen, the preliminary results show that I can probably knock that down to 15 cents per line. And if I can knock down the cost of line fixation to 15 cents per line, I can create a whole lot more lines. And if I can create a whole lot more lines, I can increase my selection intensity, which will also increase my rate of genetic gain. And then 10 minutes, good. And then lastly, we want to make more accurate selections. Okay, so we want to make sure that we are, when we evaluate material, and I pick it to be advanced or to be crossed, I want to have confidence that I'm not making an error, that line really is genetically superior than the other lines that I could have been picking. So this is, I won't go into too much detail here, but this is the redesign of the breeding program that we've put together. It looks markedly different from the pedigree selection breeding program that we had before. And so here we make crosses. Just to give you an idea of the figure, I think of a breeding program as a funnel, right? You start with a lot of material and you funnel down to just a couple. And so what we want to do is we want to widen that funnel and we want to shorten that funnel. And so in our funnel, crosses go in. We have that RGA greenhouse where we self down to fixed plants really fast. We have this one step we call LST. It's unfortunately named, but it's basically a seed amplification. So we look at panicle rows, we amplify the seed, and then I get those lines into a yield trial. And so by year 3 or 4 here, I've got lines in a yield trial and I can evaluate them for all sorts of different traits, including grain quality. I can look for yield. One of the things that we do here, and this is where the molecular breeding piece comes in, we can deploy marker-assisted selection more effectively in the breeding program by doing marker-assisted selection in this here, in this, it's not really a yield trial, in this seed amplification. And the beauty of marker-assisted selection is the heritability of a marker is almost 1. I mean, you have to give some error for genotyping error, but if it has the marker and the marker is well aligned with the phenotype, then great. I can survey that much easier than I can look at other traits. And so this is going to help us make marker-assisted selection, helps us make more accurate selections. We're going to use genomic selection in here too, and I won't go into the details like I said. One of the things that's really important though too, and I think this is how we're also increasing our selection accuracy, the strategy at IRRI, so IRRI has a unique problem compared to national partners. Our national partners have a very specific mandate. If you're BIRI, your job is to breed rice for Bangladesh. If you're IRRI, your job is to breed rice for planet Earth. And that's a much larger targeted population of environments. So what IRRI has historically done has been breed in the Philippines and hope for the best. And we do some testing of extremely late material later to see where what we've created could do well and might not do well. But we're not actively selecting for performance in those environments. We're actively selecting for performance in the Philippines, which may not be correlated. If you're a rice variety that does well in the Philippines, it's not surprising that you may not do well in Bangladesh or in India or in Cambodia. So what we're doing now is we used to have these yield trials where we have the amplification, we have our stage 1 yield trial. Then we had a stage 2 yield trial, what we call an advanced yield trial, that used to be conducted only in the Philippines. What we're doing now is partitioning the world into distinct what we call breeding zones. And those breeding zones, the technical definition of a breeding zone is a region of environments where genotype by environment interactions within the zone are minimized and between zones are maximized. And so as long as we're making progress, we're making genetic gain within a breeding zone, every other environment in that breeding zone should benefit from that genetic gain. So what we're doing now is we're sending out breeding zone specific yield trials. So the lines that I select to do well in Bangladesh might not be the same lines that I select to do well in the Philippines, or that I send to Indonesia, or that I send to Cambodia. And the idea is that these yield trials then, we will be able to create locally adapted varieties. So the model would be, the big funnel here exists at IRRI in the Philippines. New germplasm goes out into some of these other breeding zones. I used India and Bangladesh as an example. And then data comes back in from those lines into the yield trial where we want to develop estimation sets. This is more the genomic selection piece. I want to take the material I'm creating at IRRI, and I want to predict its performance in Bangladesh without testing it there. I just want to genotype it and figure out how well it'll do. And this framework is going to be what allows us to do that. That's one entry point for genomic selection into the new breeding strategy. But this regionalized focus, I think, is going to help us make more accurate selections for the regions for which we want to have impact. And part of the problem is you have to make Sophie's choice. You have to say, well, I'm going to breed for this region and not for that. And then the people to whom you're accountable will say, well, hold on. There's impoverished people in these other countries too. You need to breed for them. And I'll say, great, the cost of a breeding zone is about $200,000 a year. Where do you want us to get the money? Because at the end of the day, if we take the work that we're trying to do and dilute it in order to have global impact, we end up having no impact at all. Or worse, we have the illusion of impact without actually making impact. And so I'm much more in favor of saying, hey, you know what? We're going to do it here. We're going to do it well. We're going to show that it works. We're going to have impact. And then we're going to use that as a model to have impact in other areas and use that as a mechanism to drive investment decisions and say, hey, this investment worked here. We have reason to believe it'll work here too. Let's see if we can get people to support it. Okay, so to get into marker-assisted selection a little bit, as we close up here, there's basically two kinds of traits, if you can think about them in this sense. We have quantitative traits like yield and height, like we've talked about. There's a continuous distribution. There's not any clear binning of the traits. And then we have what we call qualitative traits or discrete traits. In rice, we have a wealth of these. A lot of them are disease traits, but basically it is if you have the gene, you're resistant. And if you don't have the gene, you're dead to the disease, for example. Whereas this is more of a scenario where you have multiple genes that are all interacting on the phenotype. So this is the kind of trait that's more amenable to marker-assisted selection, and this is the kind of trait that's more amenable to the blue side of that equation, right? So with marker-assisted selection, the two things that are super important here is, number one, making sure that you're picking the right locus. So I need to know if I say, for example, that a gene is, that this gene is important, and I do, people come up to me all the time, oh, you should use this gene. It's great. If I don't know that that gene works in the breeding zones that I care about, then we need to validate that locus first. And it's easy to think about disease in this context. You can imagine that a resistance gene in the Philippines might work, but the pathogen population in Bangladesh might be decidedly different, and those same resistance genes don't work. So you need to make sure that you're picking genes that will work in the zones you're interested in. We have put together what I call trait packages. It's sort of a term borrowed from industry. But the idea is here that we have packages of XA genes for bacterial leaf blight. We have packages of PI genes for blast. Those packages, when assembled in a single genome, will deliver resistance to all of the areas that we're interested in. And once we know that, now we can have confidence that this particular locus is going to work well for us. This is the easy part. This is the part rice does really well. We find genes better than anyone else I've ever met. And what we need to do now is then deploy markers that we can use as a proxy to select for these genes, because oftentimes the actual gene is not well known, or it's not, the functional polymorphism is not mapped. So locus validation is what I call this. This is discovering the locus and making sure that the gene does what people think it does. The part that's often skipped, and the part that makes marker-assisted selection complicated, is marker validation. There's a million different markers that you could choose in that region that you could use as a proxy for the trait, and not all of them work the same. And so, just as a token example, most of the time when we're discovering genes, that's based on linkage. And I won't go into the details here, but those of you who are interested in plant breeding, we can talk about linkage mapping. When you're developing a marker, it's based on linkage, but more than that, it's based on linkage disequilibrium in a gene pool. And so these two markers here, SNP1 and SNP2, are decidedly linked. They're right next to each other in the genome, but they are not in linkage disequilibrium with each other. And so when you think about LD, you can think about it as, if I look at the allele of this SNP, can I predict the allele of that one? And in this example, you can't. right? Because if I look at this and I say, oh, there's a C there, well, I don't really know if on the other SNP there's a C or there's a T. But if in my gene pool, if in my breeding program, this galaxy, if everywhere I have a C at this SNP, there's also a T at that SNP, and there's never any exceptions, then those two markers are in perfect LD, and one will predict the other, and those are the kinds of markers you want to deploy for marker-assisted selection. So we've done this because we develop a lot of markers, but we don't validate the markers very well. So we've done this. This is a QTL region on chromosome 1 for salt, and here are all of the different markers that are in that region, and this is plotted against what we're calling the false positive rate and the false negative rate for that marker. So if a marker says this line has the tolerant allele, but the line actually doesn't, that's a false positive. If the marker says this line does not have the tolerant allele, but it actually does, that's a false negative. So if you look across this region here, there's lots of different markers we could choose to use in the breeding program, but some of them have decidedly high false positive or false negative rates, and some of them have an error rate of 0. So what you want to do is select one of these SNPs that has a false positive rate of 0 and has a false negative rate of 0. And if that's true, then you can say that that marker is in perfect linkage disequilibrium, or LD, with the gene of interest. And that marker for your breeding program will be predictive. But if I take that marker and move it to a different breeding program, it's a different gene pool, they have different frequencies of alleles, there's no reason to believe that marker would also work. And so it is important. People sometimes invest in markers. Markers are a dime a dozen. I don't really care what marker we use. I don't care if I'm using one marker and IRRI is using a different marker. That's OK if we're targeting the same locus. And so once we've picked markers that have low error rates, then the next question becomes how useful are they in the breeding program? So let's use our XA21 as an example. It's a bacterial leaf blight gene. If you have it, you're alive. If you don't have it, you're dead when it comes to the exposure to this pathogen. Even if we have a marker that perfectly tags the resistant allele, and I can predict whether or not a line has the resistant allele, if that allele is at a frequency of 95% in my breeding program, meaning most of the material already has it, then there's no need. I don't need to invest in that marker. I don't need to invest in that locus. If the utility of that marker is high, meaning it's at very low frequency in the breeding program, then I would want to invest heavily in it. So that's another example where even if we have a marker that has a low false positive and false negative rate in the Bangladesh breeding program as well as in ours, if in my breeding program the frequency of the allele is already nearly fixed, I don't need to worry about it. If in the Bangladesh breeding program the frequency of the allele is very low, then they should worry about it, right? And it's OK that those two things are different. Sometimes we feel like we have to be doing the same things all the time. So these quality metrics are an important way, I think, to pick these markers to make sure that you're making more accurate selections. So this is my last slide. It's 1:10. We're finishing up. Well, it's not. Maybe it's not my last slide, but it's close to it. The moral of this story is just use the right tool for the right job. There are lots and lots and lots of traits for which they're controlled by very few genes, or they're very easy to phenotype, and you can either phenotype them directly, or they're very amenable to marker-assisted selection. There are other traits that are controlled by very many genes, or traits that we really can't effectively phenotype at all. Those are not traits where we should be trying to deploy marker-assisted selection. It doesn't stop us from trying, but we shouldn't be doing it. It's a waste of resources, and those resources could be used better on the blue side of that equation. So make sure you're using the right tool for the right job. So OK, so just to finish up, that's a big picture overview of some of the innovations that we're building in the IRRI breeding program that we believe are going to increase our rates of genetic gain. Now what we want to do is teach a person how to fish. And so this I think is my last slide, where we have gone through, we're working with different national partners in Eastern India and in Bangladesh in particular, and we hold what we call these TRB workshops. And these TRB workshops, I'm going to one actually next week, so we did one in Bangladesh, we did one in Eastern India, we did another one in Eastern India, and then in 2 weeks, I'm going to go back to Bangladesh and I'm going back to India to deliver another one of these workshops. And the whole idea is to just sit down with breeders in a pragmatic way and say, OK, here are the things that you can do today that will increase your rates of genetic gain. Here are the things that you can do in the future that will secure those rates of genetic gain. And we're challenging each of these national partners to put together a 3 to 5 year plan as to how they're going to transform rice breeding within their institute. And we're following up with them to make sure that those plans are sound and solid and working well. So now we're teaching these programs how to fish, more or less. You can think about it like that. And I don't want to be presumptuous about that. They're teaching us how to fish too. And the RGA example in Bangladesh is a perfect one, where they've created innovations that we're leveraging. So as a community, we're developing this strategy, but what's important is that the whole community is coming around it and more or less unifying their efforts. Once that's done, then the tricky part, which is teaching a person how to teach a person how to fish, comes into play. And that's going to be in more or less cementing these strategies at these institutions and building in people in those institutions that understand them well enough to innovate them as the landscape continues to change. And the hope is that we can then begin to come together as a unified community. With that, I mean, there's just all sorts of people that have helped me develop the program and helped me generate and analyze a lot of this data. We're funded primarily by the Bill and Melinda Gates Foundation as well as IRRI. Gregor Mendel and Charles Darwin also deserve to be on my acknowledgement slides. And we'll close up there. I'm 4 minutes late, but I'll take any questions.